Okay, thank you. We are going to begin with some background about ocean color. And we prefer the name or we prefer to use the term visible spectral radiometry because it more closely defines what is actually taking place when we make what is popularly known as an ocean color analysis. So the preferred name is visible spectral radiometry, but it's something that the name ocean color goes back to the beginning of the field and would be impossible to do away with it now. People notice that the color of the ocean changes from place to place. These are 3 paintings made around Bermuda by famous American watercolor painter. And you will see that sometimes the water is green, sometimes blue, sometimes turquoise, depending on what's in the water and what's on the bottom when the water is shallow. So we would like to be able to understand this a little bit more. And it helps us if we think of the ocean ecosystem as a machine or a thermodynamic system. So the ecosystem is an open system, and a dissipative one means it's wasting energy all the time. That energy is ending up as heat dissipation. And to keep things running, there has to be a continuous source of energy. And of course, this energy is the sun. But it's not enough to have a sun as an energy source. There also has to be some way, some means to capture the energy that is coming in from the sun. And in the ocean, the way that the ecosystem captures the energy is through the pigment molecules contained in phytoplankton. Excuse me a second. Marie, that's those gardeners outside, I think, cutting the trees. I think if it goes on forever, it will be a bit of a mess. Yeah, okay. So there has to be some way for the ecosystem to capture the sun's energy. And the mechanism, or the transducer, if you wish, are pigment molecules contained in the chlorophyll pigment molecules contained in phytoplankton. So this is an absolutely fundamental part of the how can we say, the whole marine economy. And by virtue of the light coming in and the capture by chlorophyll pigment molecules, it means that in the ocean we can have a biogeochemistry, whereas otherwise we would have a rather sterile, just a geochemistry. And as a consequence, because of this relationship between the pigment molecules in phytoplankton and the light, there is a corollary that the light escaping from the ocean, you'll see, is the basis of the ocean color signal. It carries somehow latent information about ocean biology. And this is what we want to try to discover. So this is the point. You can see here, this is a picture of the north of Scotland. You can see here that this is a feature in the color of the water that we would like to understand. We suppose that the optical signal coming from here contains the information necessary for us, so long as we're clever enough to decipher it. And we believe it's related to the presence of phytoplankton, which are individually microscopic, and in other words, invisible to the naked eye, even if you have one right here. But their collective impact is such that they're easily visible from space. So this is what we have to try to cope with. So first, to review some properties of phytoplankton. Single-celled, microscopic, and the size of the cells will be from less than a micron up to more or less 250 microns. And this is a wide size range, and if you expressed it in terms of volume, it would be an enormous size range. So phytoplankton are, for the most part, green plants. They contain chlorophyll. They grow by photosynthesis. They live mostly in the illuminated layer, but they occur everywhere and at times in great abundance. And their presence is such that they can control the color of the water, and we can detect those and quantify those color changes in the water from using sensors carried on spacecraft in orbit around the Earth. So one of the things they do is absorb light, and so they tend to modulate the way that the ocean is heated. They consume carbon dioxide. It's part of photosynthesis. And therefore, they have a major role in the carbon cycle and its relevance for climate. Their collective metabolism, scaled up around all the world's ocean and throughout the year, is absolutely enormous. The flux of carbon through phytoplankton on a world scale is 50,000 million tons per annum. It's a huge carbon flux. And the final thing I want to tell about the phytoplankton is that they are slightly heavy with respect to water, so with respect to seawater, so that there's a small tendency for them to sink at a rate that depends on their diameter. Okay. Now, the other thing to notice is that optically, and this is in the visible part of the electromagnetic spectrum, absorption of phytoplankton or biphytoplankton is highly spectral. You can see the very strong spectral structure in the absorption spectra of various phytoplankton groups. So all these things are going to bear on the basis of ocean color, as we're going to discuss. So by way of example, if we were to look at the color of the light inside the ocean at the depth where the light has been reduced to 1% of its value at the surface, as some of you may recognize that recognize this 1% light level as the base of the euphotic zone, but it doesn't matter whether you think of it like that or not. Just think of if the light at the surface is 100%, inside the ocean it becomes darker and darker with depth, and eventually we'll get to a depth where there's only 1% of the light left. The color of that light will be quite different depending on the quantity of chlorophyll in the water. So here, for example, is a scale of low chlorophyll to high chlorophyll, and the color of the light at the 1% light level will be quite different depending on the chlorophyll concentration. So when the chlorophyll is high, it will tend to be green, and when the chlorophyll is low, it will tend to be blue. So if you try to summarize the underwater light field, it's a complex interrelationship between the physics and biology, because the energy coming in at the surface has a spectral distribution, and this, as we've seen, the light under the water has a spectral distribution. This changes the way the surface ocean is heated, and therefore changes the stratification, mixed layer depth, changes the primary production and the vertical distribution of phytoplankton. This feeds back into the spectral light field itself. Spectral light field forces the vertical profile of primary production, and this has an effect on the community structure, and whether there'll be any vertical partition of taxa, and all this will again feed back on the spectral light field inside the water column. So it's a complicated story to analyze in detail. Now, ocean color remote sensing was conceived principally as a method to produce synoptic fields of phytoplankton biomass indexed as chlorophyll. It means in the units of chlorophyll concentration. So chlorophyll concentration is one way, it's not the only way, it's one way to index the abundance of phytoplankton at a particular time and place. If you want to calculate primary production, it turns out to be a very good way to index phytoplankton biomass. So that was the original objective, and this would have been something that was in the air around the beginning of the decade of the 1970s. People were thinking about this. So chlorophyll concentration is probably the most fundamental property of the ocean ecosystem. For the reason I mentioned earlier, it's the transducer between the solar energy source and the ecosystem itself. And it is being designated by the United Nations Framework Convention on Climate Change been designated as a so-called essential climate variable. That means it's something that should be studied to sufficient, or with sufficient quality, that long-term records can be used in the study of climate change, which is essentially the purpose of this entire course that's organized by Marie. So this is one of the list of essential climate variables. And this particular map shows the ocean color of the coast, eastern coast of Canada, and I think any marine scientist would agree that this is a rather beautiful picture. However, even though they're nice to look at, we should never forget that they are based on strict physical theory, in this case radiative transfer theory, and so they're not just pretty pictures. They contain a great deal of information, provided we are clever enough to interpret the information. Now, if we look at what is possible to learn from this radiometry, we have first the so-called primary products, which can be derived directly from the radiometric signal. We have secondary products, which are deduced from the primary products with other auxiliary information. And then we can make scientific applications of both of these kinds of products to address scientific issues. And also, we should never forget the societal benefit areas where there is an interplay between research, oceanography, and operational oceanography. If you look for an analogy and you go back, say, 100 years, or about 100 years, you had the science of meteorology, where the people were trying to understand how the weather works. Was a discipline of physics, but around the middle of the last century, it became highly operational and became interpreted more as weather forecasting. So it was the emphasis was no longer on the research side of meteorology, but on the operational side, forecasting, which has been developed to a fine art. So at the moment, oceanography is in this transition from being a research science to at least partially an operational activity. Now, this is what happens when we make a ocean color mission. So there are two elements in the above the ocean there that you can see. One is the sun, because the sun is the energy source, not just for the ecosystem, it's also the energy source for remote sensing of ocean color. So ocean color remote sensing is a so-called passive remote sensing. It works when the sun is out. It doesn't work at night. It doesn't work under heavy cloud. It only works by virtue of the presence of the sun. That's the first thing. Then we have the sensor, which is looking at the sea and capturing photons that are going from the sea towards the sensor. Here is an example on the black line. This is an example of a photon stream from the sun, enters the ocean, has some interaction with the water inside of the ocean, and somehow finds a way out to the surface, or back to the surface and out and to the sensor. So that black line is the ocean color signal. So some photons have come from the sun, they've gone inside the ocean, they will be modified inside the ocean by the optically active substances inside there, and they will get back to the sensor. That's the ocean color signal. That's what we have to try to interpret. But now look what else is going on. Look at all these red ones. Here is a photon stream. This one is now scattered in the atmosphere, again, reflected off the sea surface, back into the sensor. This is contamination. Here is another one. It's contamination. So all these orange ones are the photons that are finding their way into the sensor that contain no information about ocean color. They tell him more about the atmosphere than about ocean color. So one of the problems, and I'm not sure who it is who's going to talk about it, one of the major problems in this field is to remove the effect of all these orange colored paths. You know, this is the so-called atmospheric correction problem, and it's that's more than 90% of the signal that will get to the sensor. So the signal that we want is only a small percentage of what actually goes in. Okay, now I want to, oops, not what I wanted to do. I want to show you a little bit more about this part after one slide. First, I want to show you, this is what the solar energy input is to the ocean. It's the so-called solar constant. It means what is the sun delivering to the surface of the water per unit time. And you can see here the visible is this bit. That's the part we're interested in. There is a big infrared tail and an ultraviolet shoulder. Now in the visible, you can see that there is one major peak around 450 nanometers. So and then there are various people have published on the shape of this But certainly there is a consistency on what this solar constant is like in the visible. So that's the part that interests us now, is going to be incident on the sea surface and what happens when it gets to the water. So look at the left-hand one first. There are two principal processes that happen. One is absorption of photons. So if a photon is absorbed either by the water or by the chlorophyll molecule, that photon has disappeared from the light field. It's just not there anymore. The other thing that can happen is it can be scattered. Scattered means any process that will change the direction in which the photon is moving. So it can be deflected or scattered or diffracted. It implies a change of direction. Now look at the right-hand panel. You can see here is an interaction with phytoplankton. That could be absorption or scattering. Here is an interaction with the bottom. If the bottom is sufficiently shallow, then there may be some reflection from the bottom that it will be visible at the sensor. Of course, if the bottom is very deep, there's no chance that you will see the bottom in the sensor on the spacecraft. Now this one here is an interaction with molecules that are organic and dissolved in the water. It's like colored material, often of biogenic origin. Sometimes this is called yellow substance. Here is an interaction with the water itself. Water molecules can affect the paths of photons. And finally, this one is an interaction with inorganic suspended material. So there are various kinds of, you can think of them as contaminants or of suspendoids or of material dissolved or particulate in the water that can affect the signal that is seen by the spacecraft. And for the signal to get out, usually there has to be involved some kind of scattering. And in particular, the so-called backscattering. So if you think of the beam, the incident photons as having a particular direction, which we can call the forward direction. If the change in direction of scattering is such that they're now going backwards, then they might have a chance to become part of the ocean color signal. So this backscattering, which is written with a little b far back there, is turns out to be an important property of this whole story. So we call absorption A, we call scattering B, and we call backscattering B with a little b. That's the way it is. Now, in this story of scattering, there have been some prominent physicists involved. The first one on the top left is Irish. He is John Tyndall, and it was in a previous century to the others, and he was one of the first scientists to be aware of the phenomenon of scattering in seawater. He wrote about it, but he didn't really quantify it. Now this chap here was the most famous physicist of his day, is Lord Rayleigh, a very famous physicist, president of the Royal Society, and a Nobel Prize 1904 for the discovery of argon, and also an expert on scattering. In fact, scattering in the atmosphere is called Rayleigh scattering, okay, after this man. And then the third one is an Indian, C.V. Raman, who you'll see what he did in a minute. So if you look at the this picture here, this would be something that Tyndall would have observed. You have just there, can you see, a narrow collimated beam of light, which is incident on this cylinder of water and some mineral particles. And you can see that as this collimated beam, or what started out as a collimated beam, propagates through the water, it spreads out because of scattering. Okay, can you see? It spreads out in this cone of scattering. So that was what Tyndall would have been aware of. Now you can see an analog of this experiment here. This is in the real world now, in the Gulf of Alaska. This is an oceanographic front. And on one side of the front, the left side, there is a highly scattering water mass, making the water turquoise. On the right side of the front, of course, that is whatever is causing this is absent from that side. And what's causing it are these phytoplankton cells, which are made of calcite. I'll show them a little bit better soon. So here is an example from the English Channel of the same kind of thing. This is now an ocean color picture. Okay, you see this is a bloom of the same organism. And this is, if you're smart, you'll recognize where Plymouth is on this map. Okay? Now, this is what the organism looks like. This scale here is 2 microns. So it's a very small cell. Its name is a coccolithophore. And the particular feature of this organism is it's covered in shell in these plates made of calcite. And these plates tend to fall off. The fancy name is lith. And when you have a lot of these liths loose in the water is exceedingly scattering, and you get that milky color that we saw earlier. And if you wait long enough in geological time, then these coccolithophores also make make you something like the White Cliffs of Dover. That's where this thing comes from. Okay? Now, what happened? There was a famous argument. Lord Rayleigh had said that the water in the sea appeared to be blue because the water was a mirror for the sky. No more explanation required. This is very strange because, as we've said, he was an expert in scattering. This man, C.V. Raman, who also won the Nobel Prize 1930 for discovery of Raman scattering, he was not convinced by Raleigh's assertion, and he said it couldn't be like that because he'd made an experiment in which he checked the color of the water when the reflected part was removed or blocked, and he said it's all to do with scattering, and the scattering could come from different parts of the water and the contents, including the molecules of water themselves. So this he sent off his paper. It was published in Nature, and ever since, I think we can agree that Raman won the day with this view. So scattering we have to pay attention to as well as absorption. So here now, I want to make an important distinction. So please follow. Here, this is again southwest England. Here is a coccolithophore bloom. Now, can you see here this pink color? This is the Bristol Channel, and the pink color is coming out of the channel, and our geological expert here, Thomas Jackson, tells me this is red clay. So you can see it's pink. Now, such a picture is a so-called true color image. In other words, based on the wavelengths available on the sensor, this picture is as close as we can manufacture to what the human eye would see. It's sometimes called true color, sometimes called pseudo-true color. And those kind of pictures can be useful. However, we deal with another kind of image mostly in ocean color work. And this is the difference I want to show you now. We have, for the same scene, we can have true color and we can have false color. Now this is a picture of the Persian Gulf and the Arabian Sea, north. This is the true color image, similar to what I just showed you. You can see a little bit of color here and a little bit there, but mostly it's rather boring, as you can see in this image. However, when the image is processed using the ocean color algorithms, it's possible to find a lot of detail about the chlorophyll field, as you can see here. So this is the chlorophyll field mapped in false color. So There's a color bar there, but it's very rudimentary, I agree. But you can just get the idea. The red, yellow, orange colors are high chlorophyll, and the blue and green and indigo colors are low chlorophyll. So when the same signal is processed in a different way, that's when we get the information about chlorophyll. So how is this done? In the first instance, it was done by direct calibration against in situ samples. So here is water-leaving radiance spectra measured in the ocean for different chlorophyll concentrations. And you can see that these, according to the concentration, these spectra cross over each other. And according to the sensor that's being discussed, we can look at the wave bands. There's one, there's another. Wave bands that sensor has available. So this is the first attempt to have an algorithm. You can see it's logarithmic on both axes, where the ratio of reflectance at two different wavelengths was plotted against the pigment concentration. And the ratio is being used because of the way these spectra cross each other according to concentration. And you can see that on a double log scale, this is not a bad agreement with the so-called reflectance ratio. To my knowledge, this was the first such demonstration. And this was the first picture, propaganda picture, if you wish, for the distribution of chlorophyll at large scale, eastern seaboard of North America. So this is phytoplankton, and that means chlorophyll, and temperature patterns. So this is Nova Scotia, and this is the Gulf of Maine. And this feature here is Georges Bank. I'm going to show you that in a minute. Georges Bank is an important, it's a very shallow bank, important for fisheries, and was the cause of a territorial dispute between Canada and the USA that was settled by the International Court based on scientific arguments. And here you can see, let me tell you first, this is the north wall of the Gulf Stream, and this thing here is an eddy of the Gulf Stream, a warm core eddy, which has been pinched off the Gulf Stream and is now on its way to possibly land on the continental shelf of Nova Scotia. Anyway, this level of detail in the chlorophyll and temperature fields was unprecedented. For this Georges Bank, I show you what we used to have to do. The way that you would get the chlorophyll field was by making these point measurements of chlorophyll on these transects, and then you contour the data and try to construct the chlorophyll field. So the progress between that and this is remarkable, and at the world scale, it was just a revelation for biological oceanographers, or oceanographers in general, to have access to such data. And we got, I say the data became generally available to working scientists only in 1986. That's when all the data went into the public domain. Now, are we going on too long? What time do we have to stop, Marie? Oh my goodness. Okay. So I want to tell you that the optical properties of the ocean, whether it's the seawater, the phytoplankton, the dissolved organic material, or the inorganic material, their optical activity is always spectral. And so we have to be aware of the wavelength at all times. And we talk about the reflectance as a function of absorption and backscattering. Reflection in this, reflectance in this case, is not specular reflection, but is the ratio of the energy leaving the seawater to the energy that's going in. And we distinguish between case 1 waters, so-called, and case 2 waters. Case 1 water means water where the only thing affecting the color besides the water itself are algal cells and their derivatives, local derivatives such as degraded pigments and colored metabolites and so on. So that's the case 1 waters. Case 2 waters are more complex, and they contain, for example, sediments, material derived from land, colored material derived from land, and any anthropogenic inputs. So case 2 waters are typically coastal and optically complex. Case 1 waters are more open ocean. And one way to represent these effects is on this triangular diagram, where the vertices of the triangle imply, in this case, control of ocean color 100% by phytoplankton, 100% by sediments, 100% by colored dissolved material, and points on the interior are intermediate cases, with case 1 being near the top of the triangle. This figure was introduced into the literature by Schuba in her doctoral work. Now, I showed you one algorithm for the early algorithm for ocean color analysis was a straight line on a double log thing. A more recent one, where now there are more than 3,000 observations, is this rather sigmoid line as opposed to a straight one. And the idea here is that according to the chlorophyll concentration, you would choose which bit of this sigmoid curve, bottom, top, or middle, you would choose which one of those to use for the algorithm, and you would decide that by the magnitude of this remote sensing reflectance ratio, whether it was 443 nanometers or these others, always normalized to 555 nanometers. So the algorithms in use at the moment they shift according to chlorophyll concentration, and that's decided by the magnitude of these reflectance ratios. So if you look at these reflectance spectra, this dotted line always in each one is the spectrum that you would get only from seawater. Where there is plankton only, case one thing, the reflectance will be typically like this. If the dominant suspensoid water is dominated by sediments, then it will tend to look like this. And if it's dominated by yellow substance, then you will have these tails coming in like that. So we know what the reflectance spectra grossly will look like for different kinds of water. And then sometimes we want to find out what was in the water by looking at the signal. So here we could, if we know what's in the water and we know the optical properties, we can make a model that should tell us what the optical properties should be. And then those optical properties would show up in the reflectance ratio. Or alternatively, we can have an inverse model that will tell us if we know this, and we know it because we measure it, can we infer from that what the constituents of the water were. So this is one of the big problems in ocean color science. I think Bob Bruin's going to tell you about that. So here is an example where this is now a much more complicated processing than I've showed you so far, where with contamination by sediments in the North Sea, that we try to get retrieval of chlorophyll through these inverse methods. Now, I quickly want to tell you some other bits we can do with ocean color beyond measuring chlorophyll concentration. There are other pigments that we can measure, and there are other things that we can do. So both the cell size and the pigment composition modify the optical characteristics of phytoplankton. Here's an example we've seen already. This is Newfoundland, Canada. It's a cold and place, not the place you expect to get turquoise water. But when we have these coccolithophore blooms, these turquoise water will come up on the coast of Newfoundland, and the people phone up the radio station and say, what's going on, you know. It's an example of where we can identify a particular phytoplankton group just by inspection of the ocean color image. Generally, different phytoplankton groups such as diatoms and chlorophytes and so on have different absorption spectra. This is what they look like in bulk, and this is what they look like when their pigments are extracted and so on. So here is what we can get. These are samples from the Arctic, and when the sample is separated into the Primnesiophyte group here and the diatom group there, you can see that the absorption at 443 nanometers as a function of chlorophyll, these two phytoplankton groups separate out very well, and it leads us to, or it led somebody to develop an algorithm that would tell for any pixel in the image, would diagnose the probability that that pixel was dominated by diatoms. So this was published by Schuba, 2004. Here you can see spring and summer for the same place, Eastern Canada. This is spring bloom. You see all this red stuff means that these pixels were dominated by diatoms. In the same place in the summer, no diatoms here. Conversely, they're now over here. Why? Because in this Labrador Sea, the spring bloom of phytoplankton actually happens in July, because otherwise it's covered in ice. So July and even early August, the spring bloom will be here. So that's why the diatoms are now over there. And this color scale means where it's red, there's 100% probability that these were diatom pixels. So this can have great ramifications for ecology, actually. Now, another thing that we can get, without going into the details, because I think Bob Bruin will tell you, we can find the size classes from ocean color data, percentage distribution of phytoplankton by size, either in the picoplankton range, nanoplankton range, or microplankton range. It's based on based on mathematical modeling, really. So, just about finished now. So I want to summarize applications of ocean color data. We get synoptic fields of phytoplankton biomass. And why is that useful? Because a lot of people need it, for example, for initiation or validation of ocean ecosystem models. And we like to have it as a basis for calculation of prime production. We're going to show that tomorrow. It's a general tool for extrapolation to large horizontal scale, where we have only limited data on ecophysiological rates, and as a basis for study of feedback between the biota and the physics, especially mixed layer physics, and to develop indicators of ecosystem status for use in fisheries and other kinds of management. Ocean color is an integrating discipline and is our only window into the pelagic ecosystem on synoptic scales. It's integrating because it's able to combine so many aspects of marine science and is now relevant to many important societal benefit issues, such as climate. This is the purpose of this week's training. It's as well to be aware of the limitations as well as the benefits of the data. It's not a universal panacea, but it is exceedingly versatile and very highly cost effective. And there are many stimulating problems remaining to be solved. We get the most out of it when we combine it with other aspects of oceanography, such as in situ observation and modeling. And there's still a lot more to do to unite these different strands. The technology is still improving. For example, now, one of the rapidly developing things is that the transition to so-called hyperspectral sensors. At the moment, the European Space Agency sensor now in action has around 13, 14 wave bands, but the so-called hyperspectral ones will have more like 50 or 60, I think. So there is one thing that new kind of data we'll have to learn to use. And as a tool for ecosystem-based management, there's still a long way to go. It's a fascinating subject. We need new people, good people, to fill in what still is a world-scale shortage of relevant expertise. So that is problem is over to you. Okay, thank you.